session 4, an overview of analytical BA and BE inspections, the Analytical Compliance Program. This compliance program, entitled Procedures for FDA Staff in Vivo Bioavailability/Bioequivalence Studies Analytical, outlines procedures for FDA investigators who inspect domestic and international sites to ensure that the analytical portions of in vivo BA, BE, PK, PD studies submitted to the FDA are conducted using the highest laboratory standards and in accordance with applicable regulations. First in this session, we will hear from Dr. Gopa Biswas, who will discuss the inspection of the analytical portion of the in vivo BA/BE studies as described in the compliance program. Next, Dr. Sripal Mada will go over the inspection approach for in vitro studies that may be used in lieu of the in vivo studies as outlined in CFR 21, Part 320. And then Dr. Kara Scheibner will be rejoining us to go over the inspectional approach for analytical studies and assays that determine immunogenicity of therapeutic moieties. A little bit of background on our new presenters here. Dr. Biswas is a lead pharmacologist in the Division of New Drug Study Integrity within OSIS, within the Office of Translational Sciences. Her work involves inspections of firms conducting regulated BA/BE and GLP studies in support of pre-market drug approval applications submitted to FDA. And then Dr. Mada is currently a senior pharmacologist in OSIS within OTS. Dr. Mada joined FDA in 2008 as a clinical pharmacologist in the Office of Clinical Pharmacology, and later moved to the Office of Scientific Investigations and then Office of Compliance. Dr. Mada's regulatory experience includes evaluation of bioanalysis for BA, BE, PK, and and clinical pharmacology studies submitted under INDs, NDAs, BLAs, and ANDAs, as well as on-site inspections and remote record reviews of clinical and bioanalytical facilities across the globe. Please join me in welcoming our next presenters.
Hello everyone, my name is Gopa Biswas. As the title of my presentation states, I will be providing an overview of FDA's compliance program as it applies to the analytical portion of the in vivo studies. Here's my disclaimer. This presentation reflects the views of the author. It should not be construed to represent FDA's views or policies. Here are the learning objectives of my presentation today. We'll learn about the in vivo BA/BE bioresearch monitoring or BIMO compliance program or CP for analytical studies, implementation of the CP during inspection of analytical studies, and how to ensure compliance with applicable FDA regulations and meet the FDA expectations. This is the outline of my presentation. First, I will give a brief overview of the FDA Bioresearch Monitoring Compliance Program or BIMO CP in general and the Analytical Compliance Program. Then I will go over different sections of the Analytical Compliance Program with emphasis on the inspectional section to discuss FDA's expectations for achieving compliance during conduct and reporting of the analytical studies, followed by some challenge questions at the end. The FDA compliance programs have been developed for different aspects of FDA-regulated studies as seen in the list on the right side of the slide. That includes in vivo bioavailability, bioequivalence of BA/BE clinical and analytical studies, non-clinical GLP studies, non-clinical animal role studies, clinical investigator and sponsored investigator, etc. The morning presentations already covered the compliance program for GLP and animal role studies as well as for the clinical component of in vivo BA/BE studies compliance program. I will be discussing the in vivo BA/BE studies compliance program 7348 for the analytical component of these studies. These compliance programs represent a comprehensive program of on-site inspection and data audit to monitor all aspects of conduct and reporting of FDA-regulated clinical studies. The program has been established to assure the quality and integrity of data submitted to the FDA for approval of new products and marketing applications. The compliance program is also established to provide protection of human subjects and animal rights and welfare when participating in FDA-regulated studies or research. These are the main objectives of the in vivo bioavailability and bioequivalence BIMO research program, which is to ensure the protection of rights, safety, and welfare of human subjects participating in studies, to ensure the quality, integrity, and validity of clinical, analytical, and statistical data from BA/BE studies, to ensure compliance with applicable FDA regulations, and to identify significant deviations. The Analytical Compliance Program has been divided into following 7 parts or sections, and my talk will mainly focus on inspectional part 3 as highlighted in red in the slide. The Analytical CP 7348.004 covers the analytical components of BA/BE studies for marketing applications to cover new drugs, generic drugs, including PEPFAR, biologics, and biosimilars. The CP is developed to outline procedures to ensure that the analytical portions of in vivo BA/BE pharmacokinetic or pharmacodynamic studies submitted to CDER are conducted using the highest laboratory standards and in accordance with applicable regulations. The studies covered by FDA BIMO CP are governed by regulations found under 21 CFR 320 and follow the bioanalytical method validation guidance for industry. The updated guidance was recently published in 2018. The inspectional part is organized into the following 6 subparts, and I am going to focus only on parts 3 to 6. Subpart 3 under inspectional deals with stability. Stability is one of the critical aspects of analytical component of the in vivo BA/BE studies, and it starts with handling of the subject samples during the clinical component of these studies. The analytical site should store the study samples from receipt to end of analysis under stability conditions established during method validation. When the samples are received at the analytical site, the accompanying shipment records having detailed information of the shipped samples, including the study period, collections time, storage temperature, etc., and the temperature monitoring records of the samples during shipment should be available at the time of inspection. At receipt, the analytical site should document the sample temperature, any temperature excursion during shipment in sample receipt log. The sample accountability logs should also be maintained and should include the sample reconciliation in terms of number of samples shipped, received, and analyzed. If any sample query is made in case of any discrepancy, record of the query and its resolution should be available. It is important to maintain record of sample custody and storage during normal hours, weekends, holidays to ensure the safety and integrity of the study samples. During the analytical study conduct, the study sample should be handled according to the specifications in the study protocol, and records should show that they were handled per study protocol. If the analyte in samples is light sensitive, documentation should show if the study samples were protected from light. Sample tracking records to track movement of study samples in and out of the storage location, for example freezer, should be maintained and available during inspection for reconstruction of sample handling activities during the analytical study. The number of times samples were removed from the freezer should be covered by validated freeze-thaw cycle. During the analytical study, the access to the storage chambers for the study samples should be given only to those who need access to the samples. A sample log sheet or freezer log should have records of every sample retrieval and returns with time, date, and name of person handling the samples. The storage chambers should be connected with functional alarm system to alert the point of contact in case of extended temperature excursion, freezer malfunction, or power outage. The sample integrity should be confirmed during the period from shipping to the end of analytical study, and the storage duration should be covered by established long-term stability during method validation. In case of any event of alarm going off, records of action taken, servicing of the instrument, investigation, and corrective action should be available. For method validation stability experiments, some general considerations to keep in mind would be to make sure the stock solutions do not exceed the validated stability period. Method validation includes samples prepared from fresh stock solutions of analyte and spiked in appropriate analyte-free, interference-free biological matrix, for example, plasma, serum, whole blood, urine, and other matrix like surrogate matrix, etc. Stability samples should be compared to freshly prepared calibrators and or quality control samples or nominal concentrations. Stability samples should include at least 3 replicates of QCs at 3 different levels like low, mid, and high QCs. The results of the stability experiment should meet the acceptance criteria recommended in the applicable bioanalytical method validation guidance. The long-term stability of analyte should cover the longest storage duration of each sample from date of collection till the last analysis, including ISR. For studies with extended period of study duration, samples may be available and analyzed in batches or groups. In such case, the long-term storage stability duration will be different for each batch or group depending on the date of first sample collection and date of last sample analyzed for a given batch or group. Stability samples should be prepared in the same matrix or added additive example stabilizer as the study sample and stored at the same temperature or as study samples. Freeze-thaw stability should cover the maximum number of sample movement from the storage, and benchtop stability should cover maximum duration of samples kept on benchtop or at ambient temperature, or any special handling conditions, for example, on wet ice during sample preparation. Process sample or extract stability should be established to cover the duration and the storage conditions prior to sample analysis, including benchtop, refrigerator, and auto-sampler temperature. Stock solution stability should cover storage duration, condition of master or parent stock, and the working or intermediate stock solution. Reference standards storage and procurement records should document that reference standard stock solutions are made from reference standards that has not expired or reference standard that was recertified and given a new expiration date that should cover the duration of method validation or the analytical study. Concentrations to prepare stock solutions should be adjusted and a correction factor is used for salts, counterions, water, and impurity in the reference standard. Make sure the light-sensitive analytes and hygroscopic standards are handled and stored accordingly, and all precautions are taken to protect the reference standards. The fourth subpart in fractional deals with the methodology. For any analytical method, the precision and accuracy of the method should be established first. For precision and accuracy, make sure inter and intra-run precision accuracy assessments of the analytical method should be assessed using all the precision accuracy or PNA runs. A minimum of 3 independent PNA runs should be used for PNA assessment. 4 concentration levels, including the LLOQ or lower limit of quantitation concentration, covering the expected range of analyte concentrations in subject samples, are used for each run, and at least 5 replicates of each concentration measured in each run. At least one PNA run uses calibrators and QCs prepared from separate stock solutions, and the global PNA is assessed by including all the valid runs with acceptable calibration curves. Recovery should be evaluated with extracted samples at 3 concentrations, such as low, medium, and high QC level, compared with unextracted standards, that is analyte in pure solvent. Recovery should be consistent, precise, and reproducible for analyte and internal standard, and a recovery of analyte need not be 100%. Calibration curve and range of determination. Calibration curve should be prepared in same biological matrix and additive like a stabilizer if applicable as study samples. If 2 or more analytes are measured in simultaneous assays, the calibration curve should include all analytes, but the calibration curves for each analyte should be regressed separately. Concentration range of the calibration curve includes a blank sample, a zero sample, and at least 6 non-zero samples covering the expected concentration range in study. Limit of quantitation, including the lower limit of quantitation and upper limit of quantitation. The lower limit of quantitation of LLOQ determines the sensitivity of the analytical method, and the analyte response at the LLOQ should be 5 times the background noise. It should be identifiable, discrete, and reproducible. The upper limit of quantitation or ELOQ of the analytical method is defined by the highest calibration standard concentrations. A precision and accuracy at LLOQ and ELOQ should be demonstrated. Selectivity and specificity should be demonstrated at LLOQ for each analyte using 6 sources of blank matrix. Matrix effect: evaluate in blank matrix from 6 sources for both analyte and internal standard, and evaluate matrix factor when matrix effect is seen. Monitor analyte carryover from preceding sample with high analyte concentration. When the concentration of study samples exceed the upper limit of quantitation, the samples need to be diluted and reanalyzed. Use same matrix and additive if applicable as samples for dilution. Evaluate dilution integrity with same dilution factor as used for diluting samples with concentrations above the limit of quantitation during method validation or study. If the dilution integrity is not evaluated during method validation, then the dilution QCs should be included in the analytical runs. And run acceptance criteria should be based on run acceptance QCs and not dilution QCs. For partial validation, conduct partial validation to evaluate changes made in a validated analytical method. Next subpart is documentation. Documentation is critical and integral part of in vivo BA/BE studies and a must for a reconstruction of study activities during inspection. You may have heard FDA investigators saying during inspection that no documentation for any activity means that activity did not happen during the study. Make sure to follow all the relevant SOPs during method validation and the study and document the activities including all the deviations, investigations, and corrective actions. Make sure information meets ALCOA standard, that is, information is accurate, legible, contemporaneous, original, and attributable. Network and computerized systems are safe from inappropriate external and internal access, and all electronic data and computer systems are validated for intended use. Refer to 21 CFR Part 11 for electronic records and electronic signatures. Next in documentation is laboratory instruments those are used in study. For the laboratory instruments that are used in study, records of qualification, calibration, and maintenance should be up to date per applicable SOP. Activity log for instrument usage should be maintained, complete, and kept up to date. And any or all instrument errors and failures should be documented and reported. Handling of study samples in the event of instrument failure or malfunction during sample analysis should be recorded appropriately. Source data. As I mentioned before, source records allow reconstruction of study and all study-related activities. So make sure the data changes are traceable and documented. Make sure adequate system or administrative controls exist to prevent deletion or alteration of source data. Make sure an SOP exists to ensure that the source data are consistently organized and retrievable when needed. Each data retrieval should be recorded. Make sure there is an SOP in place to handle and document source data and the SOP is followed. Reintegration and reanalysis during study. Do automatic integration of chromatographic peaks and apply consistent integration parameters within a run. Reintegration and reanalysis should not be selective and should be justified using SOP-driven criteria. And make sure reintegration of chromatograms for QCs and calibrators do not bias the run acceptance. One of the important aspects of documentation in audit trails, softwares used for study should have audit trail feature. An audit trail feature should be enabled during study sample analysis to capture data acquisition, integration, and regression activities of study data. Adequate control over audit trail functionality should be applied to data security. Audit trail activities should support reconstruction of course of events during sample analysis. For data security, specified roles and restricted access of software should be given to acquire, integrate, regress, modify, remove, or report study data based on individual role of personnel. Make sure individual staff uses unique login and password for accessing software or data. Make sure adequate security is in place against unauthorized access to workstations and software used for the study. Make sure SOP-driven procedures and requirements for data storage and protection are followed. Ensure that the data storage, backup, transfer, and data archival activities are secure against unauthorized access, tampering, modification, deletion, or inadvertent loss. For data reporting, make sure reported data are accurate, consistent with source data. All method validation experiments, including acceptable or unacceptable experiments, are reported. All sample analysis runs are reported, and if there are rejected runs during sample analysis, reasons are provided for rejected runs. All reanalyzed samples, reassessed sample results are reported per SOP. Failed BE studies are reported to sponsor so that sponsor can report them to FDA, and all protocol SOP deviations are documented with justification. Internal and external communications. Communications related to sample analysis between analytical site, clinical site, and sponsor should be maintained and available during inspection. Records of any request from sponsor to change study data or final study report should be documented and available during inspection. And any issue with analytical method or study data should be reported to the sponsor and appropriately documented, and the impact on study data quality and integrity should be assessed. The sixth and final part under inspectional or subpart under inspectional is analysis of study samples. System suitability should be assessed, and system suitability assessment should be independent of study sample analysis and governed by SOP. System suitability is evaluated prior to analytical sample run and should not be part of the analytical run, and it should be evaluated, and samples for system suitability should be injected prior to resuming a sample analysis run after any instrument error or malfunction. Run acceptance: for each analytical run, at least 3 QC levels, low QC, mid QC, high QC, and LLOQ, to represent the expected range of analyte concentration in study samples should be included in duplicate. In order to meet the run acceptance criteria, 67% of QCs and 50% of QCs at each concentration level should be within 15% of nominal concentrations, as mentioned in method validation guidance. Re-injection of study samples should be justified, SOP-driven, and re-injected samples should be covered by validated process sample stability, and all re-injected samples should be reported in study report. Internal standard variability or drift should be monitored during study sample analysis. An established SOP should be available to determine run rejection based on variability or drift in IS response. IS variability limits should be pre-specified in SOP. Also determine if IS variability or drift affects the run acceptability of study samples, and QCs and samples with IS variability outside limit can be reanalyzed per SOP-defined criteria. For PK anomalies, PK sample reassay should be governed by pre-established SOP, and the potential reason for PK anomaly should be investigated according to SOP. Document who requests the repeat analysis of PK anomaly samples and document the reason for such request. The anomalies as well as the final concentration should be reported to FDA with reason for selecting the final concentration. Incurred sample reproducibility or ISR should include samples representing analyte Cmax and an elimination half-life for ISR. The ISR results should meet predefined acceptance criteria, that is 67% sample values should be within 20% of original sample values, and in case ISR failure, or rather ISR fails, procedures should be predefined for investigation of ISR failure. Now we'll have some challenge questions. The first challenge question is BIMO Compliance Program 7348.004, that is analytical, is designed to ensure studies are conducted using the highest laboratory standards and in accordance with applicable regulations. True or false? I'll give you a few seconds. The answer is true. Second question is, the following is not a justified reason to re-inject or reassess subject samples: sample processing error, IS variability, PK anomaly, instrument malfunction, poor chromatography, None of the above. Let's have a few seconds. And the answer is PK anomaly. The next question is, security of study data is best achieved by providing unrestricted access for all analytical software used for acquiring, integrating, regressing, and modifying study data of BE studies. True or false? Few seconds. And the answer is false. I would like to end my presentation by thanking the organizers for the opportunity to present today. In addition, I would like to thank my Division Director, Dr. Charles Bonapace, Deputy Division Director, Dr. Arindam Dasgupta, OSIS management, and my OSIS colleagues. Thank you. I would look forward to questions during the Q&A session.
Good afternoon, everyone, and thank you for giving me this opportunity. I am Sripal Mada, Pharmacologist in Office of Study Integrity and Surveillance. I'm going to present a talk on in vitro bioequivalence studies, its inspectional approach. As a disclaimer, presentation reflects just my views, and it should not be construed to represent FDA's views or policies. To outline the presentation, I would cover type of in vitro bioequivalence studies, methods used in these in vitro bioequivalence studies, analytical methods required for quantitation of analytes in the study samples following experiments, required documentation and its archival, blinding procedure used in drug products, reserve sample requirements and its storage conditions, and finally, the closing remarks. In general, these are the types of bioequivalence studies the office received and inspected. In vitro phosphate or bile acid binding studies are tested for locally acting drugs. In vitro or in situ permeability studies are tested for drugs intended for bioavailable applications, mostly the BCS class 1 drugs. In vitro nasal aerosol or spray testing is needed for nasal aerosol products intended for release of drug product from its container. In vitro release testing, called IVRT, utilize dissolution apparatus for few formulations and vertical Franz diffusion cell for ointment and cream formulations for topical delivery, and in vitro permeability testing, called IVPT, for transdermal formulations. For phosphate or bile acid binding studies, these are few drugs that were submitted to agency utilizing the binding studies. Cholestyramine and cholecebulum hydrochloride are considered bile acid sequestrants. Sevelamer hydrochloride and carbonate are polymers that bind to phosphate ions. Lanthanum carbonate, calcium acetate, and calcium succinate or salt-based molecules react with phosphate ions. Nasal aerosol or spray testing is for drugs that are intended for inhalation and nasal route of administration. The drugs administered by inhalation through the mouth must be atomized into smaller droplets and those administered by nasal route, so the drugs can pass through the windpipe or trachea into the lungs. In vitro bioequivalence evaluation for locally acting drugs delivered by nasal aerosol or spray are usually characterized by 7 tests that includes single activation content through container life, droplet size distribution mostly conducted by laser diffraction, drug in small particle for suspensions, droplets for emulsion formulations, or size distributions can be evaluated by cascade impactor. Drug particle size distribution by microscopy, and this is intended only for suspension products for visual observation. Spray pattern to see the shape of the spray, plume geometry for side view of the aerosol cloud, priming and repriming of delivery of labeled dose based on single activation content. We recommend any instruments used in these studies be operated within the manufacturer's recommendation, and any change in the manufacturer's setting will have to be supported. During inspection, the instrument setup and operational conditions will be reviewed. Modern equipment can provide plots of obscuration or percent transmission, and particle size distribution can provide D90, D50, and D10 over the entire life of single spray. The plots from these experiments need to be retained. In fact, the source documentation for all tested parameters need to be retained. Protocol or SOP should include prior criteria for defining the testing procedures. The submission should include photographs when quantitated by manual analysis, or digital images when quantitation is by automated image analysis. To be specific, images would clearly indicate plume angle, width, and height. Analytical runs include at least 3 or more concentrations of quality control samples. Recommend the test method or SOP include a standardized shaking procedure prior to any testing. The IVPT pivotal study should utilize a balanced design directly comparing the test and reference on skin from the same set of donors, each with the same number of replicate skin sections per donor per treatment group. The validity of each skin section dosed in the study should be qualified. The acceptable barrier integrity tests may be based upon the transepidermal water loss, called TEWL, or electrical impedance or conductance, or tritiated water permeation across the skin. The skin thickness should be measured and reported for each section and should be relatively consistent for all donors whose skin is included in the study. The accuracy and precision of receptor solution sample collection at each time point should be appropriately qualified. The validation of the receptor sample analytical method should include relevant qualification of dilution integrity as well as the stability assessments. So the other IVPT qualification procedures include the skin preparation procedures need to be documented. Example, the skin thickness membrane by dermatome need to be documented. The thickness of skin sections that mount on the diffusion cells need to be measured and recorded. This is mostly done by the micrometer. The skin storage conditions include temperature conditions are recorded. Duration for which the skin was frozen, it includes record of the freezer ID if the samples were stored for long period. Number of freeze-thaw cycles to which the skin was exposed, which is supported by the validation data. Documentation of anatomical location and demographics, example age, race, and sex. Control of dose amount, dosing technique, dose duration should be documented in source records. Sample handling, storage, and tracking documents need to be maintained. Ambient laboratory temperatures and humidity conditions during the study should be monitored and reported. After the experiments are completed, the samples are collected from those experiments and sent to the instrumentation lab, where the samples are quantitated for analyte estimation. Mostly the samples are analyzed using different instrumentation techniques. These are the instruments used for phosphate, calcium, and bile acid salts. The instruments include the HPLC with ion conductivity, inductively coupled plasma atomic emission spectroscopy, which is ICP-AES, or HPLC with UV detection or photodiode array detection. Or in the recent studies, we also seen the LCMS-MS methodology for these type of the bile acid estimations. And in addition, for perfusion studies or drug-in particles or droplets or IVRT and IVPT studies, basically two different kinds of instruments were used, like LC-MS method or HPLC with UV or PDA detection were used. For these type of quantitation, the instruments, methodologies, and study conditions appropriately qualified, validated, and verified. Instrument settings established during pre-study validation would be used in the study. For comparative studies, use of same settings will ensure that test and reference are studied under the same instrumental conditions. Validation of all in vitro tests for accuracy and precision prior to the study. Any changes in instrument setting and testing procedure need to be validated in addendum. This slide explains what we covered during inspection. That includes sample storage area, including the freezers and refrigerators. Storage facility temperature conditions will be checked and confirmed. Drug product receipt and accountability, that includes storage, handling, and processing. Confirmation of the reserve sample and its storage conditions. SOPs, protocols, and protocol deviations, if any. Training records of staff for part of the study conduct. Audit trail and data security features of instruments. Instrument calibration and maintenance records. Documentation of experimental conditions and sample handling, that includes the correspondence folder. Inspection of the source documents that are related to study specific include method validation of experiments, method validation of instrumental methods, and sample analysis from the experiments will be reviewed. For method validation of experiments, the parameters like the precision and accuracy, selectivity, recovery and matrix effect are few examples that are included. But if additional parameters are included to support the experiments, will be reviewed during the inspection. Sample analysis from the experiments include sample processing sheets and its procedures, sample reassay and its acceptance criteria, method performance based on the performance of the quality control samples and the run acceptance based on the calibrator will be reviewed during the inspection. Method validation for analytical methods include precision and accuracy in receptor matrix, sensitivity, selectivity, dilution integrity, stability assessments that include Bain stop, extended storage, freeze-thaw matrix stabilities, stock solution stability, and the robustness. And additional parameters, if are added in the method, will be reviewed at the time of inspection. In the next few slides, I'm going to present documentation expectation at the time of inspection. Separate reports for method validation and pivotal studies. Maintenance and control documents of study facility environment and systems. The temperature logs for freezer and facility are made available at the time of inspection. Experiment dates and information related to principal investigator, including contact details and site address, should be clearly presented in the study report. Documentation indicating who conducted the study with dates. Archival of study records, including source documents like certificate of analysis, and raw data including experimental procedures, sample processing, and chromatograms are made available at the time of audit. Turn on audit trail feature and electronic data and is secured for audit. The firm should follow good documentation practices that include contemporaneous recording of procedures, observations, and deviations to allow reconstruction of the study. Study personnel training records and qualifications that include general and study-specific training, and all documents are available during inspection. Unexpected results and deviations from protocol or SOP with justifications for deviations are documented. The original and reanalyzed data with the reason of reanalysis would be tabulated in the study report. Original chromatograms, if generated, are made available at the time of inspection. SOP should clearly indicate the objective criteria for repeat testing. Drug accountability records for receipt, storage, and use of test and reference products are maintained. A table of drug accountability from experiment would be helpful. Documentation includes instrumental output reports, and photographic or graphic material is valuable. We recommend that documents be clearly labeled to indicate a test drug product, batch number, and testing conditions. If manual actuation used in priming and repriming experiment, documentation indicating secondary analyst review and confirm the number of activations used in these experiments. However, if automated activation system is used, it will decrease the variability of drug delivery due to operator factor that will increases the sensitivity of detection potential. Also recommend the test method or SOP include a standardized shaking procedure prior to testing, following the label instruction. Blinding of drug products is always beneficial for majority of in vitro bioequivalence studies. SOP or protocol describe the procedure for sample blinding of test and reference product with different codes and its repackaging. Blinding includes labeling, sampling, and repackaging of test and reference product into identical containers, and the container details are as per the SOP. The product will have to be masked and to result in blinding the identity of drug product. Analysts performing the experiments and data evaluations be blinded to the identity of the samples. The reserve sample requirement is the applicant or the contract research organization shall retain reserve samples of any test and reference used in conducting in vitro bioequivalence study required for approval of the application as per 21 CFR 320.38 and 21 CFR 320.63. Reserve samples from test and reference products should have adequate quantities retained from each shipment. Firms' procedures allow to randomly select the drug products for reserve samples from each shipment. The reserve samples are stored in segregated area with access limited to authorized personnel and in original container with proper label. Most common deficiency or observation for in vitro bioequivalence studies include reserve samples not retained, improper handling, storage, and reserve sample retained under 21 CFR 211, which is CGMP regulations and not bioequivalence. We have two guidances for industry. One, handling and retention of BA/BE testing samples. And the second is compliance policy for quality of BA/BE samples retained under 21 CFR 320.28 under C. To summarize my presentation, the methods used in the in vitro bioequivalence studies should be properly validated prior to the study. Analytical methods should be sensitive and selective for quantitation of analyte. Follow good documentation practices that include contemporaneous recording, observation, and deviation to allow reconstruction of the study. Drug products are randomized and blinded for proper evaluation of the data. Reserve samples are selected, stored in proper conditions, and made available during inspection. Okay, now I have 3 challenge questions for the audience. The first challenge question is, which of the following are example of membrane or skin barrier integrity test conducted in IVPT studies? A is transepidermal water loss, TEWL. B is electrical impedance or conductance. C is appreciated water permeation, and D is all of the above. I'll give few seconds. Okay, the answer is all of the above. Yeah, here is the second challenge question. Which of the following documents are maintained at the study site and are made available at the time of inspection? A, contemporaneous documentation. B, analyst training records. C, instrument calibration and maintenance records. D, chromatograms and audit trails. E, all of the above. And the answer is all of the above. These are all documents required at the time of inspection. And the third challenge question is, for 21 CFR 320.38 and 21 CFR 320.63, the applicant or CRO shall retain the reserve samples of test and reference used in conducting an in vitro bioequivalence study. Is it true or false? And the answer is, of course, true. This is a regulation. I thank everyone for attending this talk. I also thank Dr. Julia Chow, Dr. Kimberly Benson, and Dr. Stanley Aup for providing me this opportunity for this presentation, as well as review of the slides. And if you have any questions, I'll be available at question and answers panel. Thanks and have a great rest of the day.
Good afternoon, and thank you for a second opportunity to speak with you all today, this time about immunogenicity studies and what to know for an analytical inspection. During this talk, I will discuss differences between analytical inspections for small versus large molecules, the relevant guidances to help you along the way, and critical method validation parameters for PK and immunogenicity assays. I will also sprinkle in some information to know for sample analysis, particularly for immunogenicity studies. There's a lot of information packed into this talk, so let's get started. First of all, are there differences in analytical inspections for small and large molecules? Well, the answer is yes and no. And I know this is clearly not super helpful, but it is honestly the truth. Many aspects of the inspection will be the same because during an analytical inspection, OSIS's main focus is to verify the integrity and reliability of the data generated. That doesn't change with respect to biosimilars. And we do this by looking at numerous aspects of a firm and the study or studies we're reviewing. Some highlights here include a review of all method validation and study records, both paper and electronic, sample management, handling, and documentation. Biosimilar studies can involve multiple methods, multiple studies, for example, PK and ADA, and multiple analyses of the same sample. Good documentation practices are always critical, but especially so here. We also look to see if all SOPs are up to date, relevant to large molecules, and followed by all staff members. A large part of data security are audit trails for any software being used to acquire, analyze, and track data. Are these active, and are they reviewed regularly? And finally, is documentation of all activities contemporaneous, accurate, and detailed enough to allow for a full reconstruction of a study? But while there are many similarities, there are also multiple differences as well. Some of the inspectional elements will be different. For example, some methods may have different validation parameters. There may be multiple methods within a single study, and there will be different equipment, instrumentation, and software. It is also likely that more of your staff will be involved. Multiple methods may involve multiple departments and personnel. And finally, the sheer number of records and data reviewed during a biosimilar inspection will be greater. Do not underestimate the time and resources that will be required. So, are there any applicable guidances to help you on your way? I'm glad you asked because indeed there are, and they are full of very useful information and resources. The first one is the Bioanalytical Method Validation Guidance from May 2018, and it's useful, specifically the sections relevant to ligand binding assays, which are used for PK assays in biosimilar applications. Second, the Immunogenicity Testing and Therapeutic Protein Products Guidance from February 2019 is relevant for antidrug antibody and neutralizing antibody assays. I've included the links to both in this slide. But now I really want to transition into some of the specific validation differences in validation parameters for biosimilar assays, because these are aspects we will certainly pay attention to during our inspections. So first, let's look at PK ligand binding assays and specifically the calibration curve. The first thing to know, and this applies across all PK, ADA, and NAB assays, samples are analyzed in duplicate wells, and the average value of those wells is reported. In this table, you can see replicates 1 and 2, and the average response of each calibration standard. The use of duplicate wells invokes the use of %CV as an additional acceptance criterion. The %CV between duplicate wells is calculated, and the acceptance criteria is less than or equal to 20% or 25% for the LLOQ and ULOQ. In this example, you can see that the 100 microgram per mil standard has a %CV equal to 25%, and thus it is unacceptable. It should be removed from the regression, even if accuracy is acceptable. Too many replicates with unacceptable %CV values could indicate an issue with the assay, and this should be monitored closely. Ligand binding assays will typically not produce a linear response, which can be seen in the graph here. Thus, regression is usually done using a 4 or 5 parameter logistical curve fit. And finally, anchor points are often used in ligand binding assays to improve curve fit. These are additional standards added at the low and or high end of the curve, but are not a new LLOQ or ULOQ. In fact, no precision or accuracy criteria are applied to anchor points, and they do not extend the range of the assay. The next parameter with differences from small molecules is precision and accuracy. At face value, P&A assessments, as seen in this table, look very much the same, but let's look deeper. First, instead of 3 P&A runs, it is recommended that a ligand binding assay have 6 independent P&A runs, as shown here for an LLOQ concentration. Data from all runs is included for inter-assay statistics. The next new concept is total error, which is an additive assessment of percent accuracy and percent CV. Acceptance criteria for total error is plus or minus 30% and plus or minus 40% for the LLOQ. This value is not always used, but it is a good way to monitor the assay. Finally, and this is no different from small molecules, but always worth repeating, in P&A runs, there are no acceptance criteria for QCs. Precision and accuracy results are what they are, failed QCs and all. A P&A run should not fail unless 1, the calibration curve is unacceptable, or 2, there is a contemporaneously documented technical or instrument error. That's it, that's all. Failed QCs do not result in a failed P&A run. For example, Run 2 had a 130% accuracy, which was unacceptable. However, these results should still be included in inter-assay P&A statistics. Furthermore, identified outliers should be included in an inter-assay P&A. If the outlier is definitively linked to a contemporaneously documented error, results can be presented with and without the outlier. Something you won't see in small molecule assays is the MRD, or minimum required dilution. The MRD is applied to all samples, CCs, QCs, and unknown samples to minimize nonspecific assay interference. An optimal MRD should yield a high signal-to-noise ratio and an assay signal close to that in assay diluent without the multiple interfering factors present in plasma. Derivation of an MRD requires assessing multiple QC concentrations in serial dilutions of matrix to ensure an adequate signal-to-noise ratio across the entire range of the assay. And typically the recommendation is for the MRD not to exceed a dilution factor of 1 to 100. The final PK parameter I'll discuss before moving on to immunogenicity is comparability. During sample analysis, the biosimilar and reference drug or drugs will be assessed in a single assay, and the biosimilar will be used for the standard curve. To demonstrate comparability at minimum, QCs of both the biosimilar and reference drug should be prepared, and precision and accuracy of both against the biosimilar curve should be assessed. The same acceptance criteria are applied to all QCs. Other key validation parameters, for example, selectivity and stability, should also be considered in determining comparability for a biosimilar assay. Let's now turn to the key differences in immunogenicity assays from small molecule assays. First, assays are semi-quantitative. There are no calibration standards and there are no accuracy criteria applied. In immunogenicity assays, precision is king. Second, cut points are used to define whether a sample is ADA positive or ADA negative. Cut points can be influenced by matrix, assay background, and assay variability, just to name a few factors. Low positive control is not predefined and is established based on the sensitivity of the assay. And finally, immunogenicity samples will likely contain some concentration of the study drug, which can interfere with the assay. The drug tolerance of an immunogenicity assay must be established to ensure reliable results. First, we'll discuss precision. ADA and NAB assays yield results of either positive or negative. And while the assays are not quantitative, the precision of an assay is critical. If an assay is not precise, determinations of ADA positive and ADA negative samples will not be reliable. Precision assessments should include runs on different days with different analysts and different instruments to account for all potential sources of variability. There should be 6 independent runs, each with 6 replicates of positive and negative controls, and the acceptance criteria for these assays is a percent CV of less than or equal to 20%. Precision should be assessed for all assay tiers, screening, confirmatory, and titer. And on this slide is just an example of data for precision assessments in screening, confirmatory, and titer assays. Moving on to cut points, which is probably the most abstract concept in immunogenicity assays. Determination of cut points for each of the 3 assay tiers is critical and fundamental part of assay validation. In the absence of a quantitative assay, cut point values are used to determine the positive or negative status of a sample and to define the titer. As such, appropriate statistical methodology is crucial. Specific cut points are determined for each assay tier. For screening cut points, use of a minimum of 50 individual treatment-naive matrix lots is recommended, as is a false positive rate of 5%. Screening cut points can either be fixed or floating, though floating cut points are more commonly used because they account for the expected plate-to-plate variability seen in these assays. For the confirmatory assay using a competitive inhibition format, the cut point is expressed as drug-mediated % inhibition of treatment-naive matrix lots. Because this assay is more specific, the recommended false positive rate is 1%, and confirmatory cut points are typically fixed. And finally, the titer cut point is often the same value used for the screening cut point because the same assay format is used. However, an alternative cut point is sometimes used to ensure reliable titer values due to variability around the screening cut point. Establishment of cut points allows for further criteria to be applied to positive and negative assay controls, in addition to the precision criteria discussed earlier. One example of a criterion is as follows. 3 of 4 negative control replicates must yield an assay signal less than the screening cut point. As you can see here, the data in the top portion of the table meets this criterion, but the data at the bottom does not, as 2 replicates are above the cut point and therefore positive. In another example, all LPC replicates must have an assay signal above the cut point. Again, the data in the top of the table meets this criterion, but the data in the bottom does not, as 2 replicates are below the cut point. Finally, all confirmatory LPC samples must have a % signal inhibition above the confirmatory cut point. And in the table on the left, this criterion is met. But in the table on the right, 2 replicates fail to meet this criterion. In a semi-quantitative assay without a calibration curve, setting positive control concentrations is not straightforward. However, setting an adequate LPC concentration is critical because the LPC plays a vital role in 1, monitoring the performance of the assay in a signal range close to the cut point, 2, monitoring assay precision, and 3, monitoring the consistency of a positive control screening, positive. And all of these things combined will ensure the reliability of unknown sample results. The LPC concentration for ADA and NAD assays is typically determined from sensitivity assessments. The sensitivity is calculated based on the concentration at which serial dilutions of the positive control cross the cut point. The LPC is then subsequently calculated from the sensitivity based on a 1% failure rate. In real-world language, the LPC should be responsible for an assay run failing about 1% of the time. If the LPC never yields negative results, it's probably not an optimal LPC concentration. On this slide, we can see data from a sensitivity assessment. The average calculated sensitivity was 4.5 nanograms per mil, and the LPC concentration selected was 20 nanograms per mil. The question is now, does the LPC actually yield a 1% failure rate? And is the LPC relevant to study samples? We saw this data before. On the bottom part of the table, we can see that 2 LPC replicates fall below the cut point. Thus, when set at a 20 nanograms per mil, the LPC can indeed cause the run to fail. In the graph pictured here, we can see that the average LPC signal, the upper line, is close to the cut point, the bottom line, and also falls well within the signals yielded by the majority of study samples. However, if the average cut point, or if the average LPC response was here, at this high upper line, it's too far away from the cut point to monitor the assay in this signal range and completely irrelevant to any study samples. It is also quite unlikely that an LPC concentration yielding this high of a signal would ever result in run failure. The final validation parameter I'd like to talk about today is drug tolerance. Immunogenicity samples may also contain substantial concentrations of the drug product, especially in longer-term multiple dose studies. Because these are binding assays, the drug product can potentially interfere with the assay to yield false negative or false positive results. Suboptimal drug tolerance may require additional optimization steps during method development to achieve the most drug tolerant and sensitive assay possible. Drug tolerance is determined by assessing positive control samples both with drug and without drug and in the presence of increasing drug concentrations. The highest drug concentration allowing detection of a positive control is the drug tolerance. In this example on the right, you can see that increasing drug concentrations simultaneously decrease the assay signal, and all concentrations above 100 micrograms per mil yield negative results in the positive control samples, and so these would be considered false negatives. Thus, these data indicate that assay results for any samples with known drug concentrations exceeding 100 micrograms per mil may not be reliable. And so to finish, we'll move on to our 2 challenge questions. Challenge question number 1, is the following statement true or false? Precision and accuracy are critical parts of immunogenicity assay method validations. This statement is false. ADA and NAD assays are not quantitative assays, and therefore accuracy is not a relevant validation parameter. And now for challenge question 2. Biosimilar bioanalytical inspections, A, involve many of the same assessments as small molecule bioanalytical inspections. B, involve different assessments compared to small molecule bioanalytical inspections. C, may involve multiple methods, method validations, and studies. D, will assess data integrity and data reliability. Or E, all of the above. And the answer is E, all of the above. Once again, I would like to thank you so much for the opportunity to speak and for your attention. I look forward to answering any questions during the speaker panels.
Thank you for the excellent presentations. We will now have our Q&A panel with all the session presenters. Our first question will go to Dr. Biswas. In cases where a sample exceeds validated stability, e.g. long-term storage, FTO or benchtop, and data is generated, is it recommended to report the data with footnote identifying the excursion, but follow up with further stability testing? Is the additional stability testing required? Should the value not be reported due to sampling being outside stability?
Thanks, Nora. Thanks for the question. It's a great question. That's a great question. Yes, that's right. The sample should be covered by validated stability, and if there is any sample exceeding the validated duration, that should not be skipped. It should be reported, and it is always recommended that additional validation for storage stability, whether it is long-term storage or freeze thaw or benchtop, should be conducted to cover the extra stability period that is not available before when you do the initial method validation. So yes, the answer is yes, you have to do additional method validation to cover the extra storage duration, and the all these data should be reported, whether they are covered by the stability or not. Thank you.
Thank you for that information. We have a few more questions for you. What are the FDA plans for implementing ICH M10? And does M10 supersede the 2818 FDA BMV?
Thanks, Nora, for another great question. I see there are several questions related to M10, and there's a lot of curiosity about it. The ICH M10 guidance or the document has been finalized in May, but it is going through FDA clearance right now before it can be considered in place of FDA guidance. So until it is finalized and released, for the studies done currently or before the ICH M10 is authorized or released, all these studies will be covered by method validation guidance 2018, and until the M10 is published as FDA guidance. And similarly, for other agencies, they will post on their sites when they adopt the M10 guidance as well. So will FDA. Thank you.
All right, thank you for that response. Our next question will actually go to Dr. Mada. Dr. Mada, for an in vitro BE study conducted using a dissolution testing method, the concerned dissolution testing method does not involve the use of any biological matrix. Dissolution testing is performed using a chemical dissolution medium. As it is not a bioanalytical method, the dissolution method was validated applying ICH Q2R1. It is the same dissolution testing method that should be used for batch release. Would you please explain why this validation was evaluated as insufficient and why the introduction of QCs within each HPLC sequence was requested while a 100% standard solution is regularly injected through the HPLC sequence as for a bioanalysis method, even if the dissolution method used is not a bioanalytical method?
Thanks, Nora, for the question. Yes, for if it is a dissolution study, just a dissolution study intended for analysis of the analyte in a solution that do not involve the biological matrix, a complete linearity will be established during the method validation. And since the samples for calibrators and QCs are same, there is no requirement of quality control samples additionally in addition to the calibrators, because you can still use few representative concentrations from the full calibration curve that was generated from the validation. You can use the representative samples, and that can be sufficient because that can generate the back calculation using the bracketing approach also. I think that should be fine.
All right. Thank you so much, Dr. Mada, for that response. We'll go to another question for you. With IVRT and IVPTBE studies being required to be run within a GLP compliant quality management system, is it recommended to follow the FDA BMV guidance strictly for analytical method validation, or is it acceptable to use different standards such as ICH Q2R1?
Thanks, Nora. This is also a great question. To be specific, there is no analytical guidance recommended by FDA for specifically for IVPT and IVRT studies for method validation or method development. But however, they can use any guidance, including the bioanalytical method validation guidance from 2013 or ICH Q2R1, as long as the method is accurate and sensitive and it can be reproducible. I think that should be fine. Any guidance will help as long as the method is properly validated.
Okay, thank you. And one more question for you, Dr. Mada. For IVPT, which guideline is to be followed for MV and sample analysis?
And the answer is same, actually. This is the same question I addressed in the previous question. so they can use any method validation guidance. And to add on this, we had recently IV-RT, IV-PT quality management workshop from Office of Generic Drugs by Sam Rene. I think that would be a good guide for use of the quality management system in any of these IVPT, IVRT testing, because that helps understand, and that will be a good guide for even for method validation part the Sam had covered. Thank you.
All right, thank you for your responses once again. We have a few questions that came in for Dr. Kara Scheibner as well. Dr. Scheibner, where can we learn more about method validation parameters?
That's a great question. And, in this short time, I really only was able to focus on a few. But for I mentioned both of these guidances in my talk, and I have links to them. For PK assays, the bioanalytical method validation guidance, specifically sections related to ligand binding assays, is a fantastic resource. And then for the ADA and NAB immunogenicity assays, the immunogenicity testing of therapeutic protein products is a go-to guidance, and they both have a ton of great information for all of the different method for validation parameters that should be considered for each of the assays.
All right, thank you for that information. Another question for you. Do you run encouraged sample reanalysis ISR studies for immunogenicity studies?
So in terms of for biosimilars in general, for PK assays, you do, just as you would during a normal small molecule assay. However, specifically for immunogenicity studies, ADA and NAB assays, you do not perform ISR, again, because these are not quantitative assays. And so therefore, ISR is not required.
All right, thank you for that response. We'll go back now to Dr. Biswas. We have a few more questions for you. Is the use of 6 blank plasma matrix lots sufficient to confirm internal standard suppression, or is it the expectation to introduce more lots to verify suppression?
Thank you for the question, Nora. As suggested in the guidance, 6 slots of blank plasma matrix should be sufficient, but it's up to the analytical site if they want to introduce more. It always, increases the confidence in the assay if you want to introduce more, but there is no harm in it. But basically, 6 blank plasma lots should be sufficient. Thank you.
Okay, and what is the acceptance criteria for blank samples?
Okay, another good question. The acceptance criteria, first of all, the blank sample should not have any, signal at analyte RT, retention time. And if there is any noise, it should be less. It should be within 20% of the LLOQ, and so it should be, in other words, it should be below level of quantitation. Thank you.
All right, thank you for clarifying that. One more question for you, Dr. Biswas. Should the stability evaluation be done at low and high QC levels as per the 2018 BMV guidance, or even mid QC to be included as mentioned in your presentation?
Thanks again for that question, Nora. No, per the BMV guidance published in 2018, the stability can be evaluated at low and high QC, but it's optional to include mid QC. That is why I just covered, I mentioned about mid QC as well. But yes, low and high QC level can be used for establishing the stability. Thank you.
All right, thank you. And now we'll go to some questions that came in for Dr. Mada again. Dr. Mada, in IVPT studies where the human skin should be considered as, or should the human skin be considered as reserved samples?
Yes, no, that is not required because human skin can be just a medium for the method, but actual samples tested on it, like ointments or creams, are considered as reserve samples, and they can be retained as per the regulation 320.38 and 68.
Okay, thank you for that response. And another question that had come in for you, or incurred sample reanalysis, is it expected for in vitro study?
Again, the answer is no. These samples are not required because you are doing the experiment freshly and the storage of samples after the experiment is not required. So as the samples are freshly analyzed, the ISR is not required.
Okay, thank you. And we might have time for one last question for you, Dr. Mata. What does properly validated analytical method exactly mean? I.e., fully validated or a non-validated method would be acceptable?
Properly validated is, again, validation to be done as per the sample handling condition. For example, the samples are analyzed and they are properly validated for accuracy, precision, and robustness. All these are tested for, especially for in vitro type of testing. But if you use the guidance, it is fully validated, where it has other components into it, like the hemolysis and lipemic and all other parameters like long-term stability. All those are not required for in vitro testing. So that's why it is properly validated and which cover all the parameters that are the sampling handling conditions are seen.
All right, thank you. As mentioned, that is all we have time for today. Thank you again to all our presenters and panelists. This concludes day one of the Office of Study Integrity and Surveillance workshop. Now we will hear some closing remarks from Brian Folian. Brian is the Deputy Office Director of OSIS in FDA CDER's Office of Translational Sciences. His work focuses on ensuring the reliability of bioavailability and bioequivalence and good laboratory practice studies to support FDA decision-making on human drug and biologic product applications. Now I will turn it over to Brian.
Thank you very much, Nora. We've heard a lot of information today, and I want to thank everyone who's remained, all the attendees and the presenters that remained on the call. It's been a very collaborative engagement. I want to thank Nora and Brenda as SBIA for the wonderful support for this workshop today. When we think about what we've heard, it's kind of, we take a step back, and it's really an excellent opportunity to pause and recognize the scope of FDA's bio research monitoring program and everything that it touches. I mean, today we spent an entire day touching on just a small fraction of that BIMA work that's overseen by ORA and our counterpart office, the Office of Scientific Investigations. In particular, it was just a light touch. A lot of questions came in about what we're doing in specific situations and specific cases, specific ways and methods are validated, and we've only been able to, in our presentations, touch on small components or a single type of method validation within across just 3 programs. The conversation today was on when you think about the fact that OSI, the Office of Scientific Investigations, has another 15 BIMO programs that are overseen by their office. It just is an impressive amount of information that we could, only just begin to present to you all. So, definitely look forward to more conversations on this. Today's presentations really are that high-level glimpse of how OSIS works to ensure the data reliability and human subject protection in the cases that's necessary for purposes of bioavailability, bioequivalence, good laboratory practice, and animal studies that we heard today that are submitted to the agency. So a significant amount of effort goes into those investigations and examinations and the work with the attendees here on the call today. We're not going to rehash everything in the close. The various clinical and analytical and non-clinical studies and study conduct considerations that were covered today was impressive. The questions were asked were great. I do think for purposes of what you heard today, the key takeaway from day one of the OSIS SBIA workshop is really these conversations about what OSIS compliance programs are looking at are really focused at ensuring that the clinical and analytical BAB testing sites and GLP and animal rule testing sites have the documentation and the information necessary to allow reconstruction of the studies conducted at those sites and to allow us to assess thereby the reliability of the studies and the study data and the protection of the rights, safety, and welfare of the subjects enrolled in those studies. And that's really what today was about, is about walking through the overall high-level view through these presentations on what we do and how we come to facilities and try to determine whether the clinical study met basic GCP expectations, good clinical practice expectations, that is, and whether the analytical studies, the bioanalytical studies, were done in a manner that allows us to ensure that the data produced is reliable for purposes of measuring BA or demonstrating BE. So really nice framework for attendees to consider for future interactions with the agency. And for tomorrow, really a piece for the attendees to come to hear more about how that is actually effectuated. It's going to be a day of case presentations that should really drive home those conversations about how we, OSIS, when we come out to do those, BA and BE study audits and those GLP and animal testing site, assessments and evaluations, how we're going to look and how we have historically looked at study conduct. And it should provide a very informative, confirmation of where the agency is, where expectations lie. So, I really welcome, continued collaboration tomorrow. A lot of good questions would be really beneficial for the agency, for OSIS, in these programs that we're discussing. So look forward to everyone coming tomorrow, and thank you. Have a good night or good morning, wherever this conversation finds you. So thank you. Back to you, Noah.
All right. Thank you, Brian, for those remarks. A few final reminders as we close out our day. Recordings will be posted on our website. You can see it listed there at www.fda.gov/CDER-SBIA. Reminder to use the evaluation link to obtain a certificate of attendance to support continuing education credits from SOCRA, RAPS, SQA, and ACRP. However, CE claim codes will be sent to attendees for continuing education credits for physicians, pharmacists, and nurses. And also the evaluation and certificate is available for 2 weeks only, so that will be up until August 3rd. And that brings our day to a close. See you all again tomorrow for day 2.